Hello friends and welcome. Today I am going to deliver a lecture on variation in toxic responses, toxicity testing methods and LD50 experiment. Now let us first try to have a look at the main objectives of our today's deliberation. Number first, to give a general and thorough idea about the drug toxicity. Number second, we shall be explaining in detail the variations and pathology of drug toxicity. Number third, to discuss in detail toxicity testing methods. Number four, to discuss at length various approaches related to toxicity testing. And finally, we shall be discussing LD50 experiment. Now, we will start with the brief idea about the drug toxicity. Toxicity refers to how poisonous or harmful a substance can be. In the context of pharmacology, drug toxicity occurs when a person has accumulated too much of a drug in his bloodstream, leading to adverse effects within the body. Drug toxicity may occur when the dose given is too high or the liver or kidneys are unable to remove the drug from the bloodstream, allowing it to accumulate in the body. Drug toxicity can occur as a result of over-ingestion of a medication, having too much of a drug in a person's system at once. This can happen if the dose taken exceeds the prescribed dose, either intentionally or accidentally. However, with certain medications, drug toxicity can also occur as an adverse drug reaction. In this case, the normally given therapeutic dose of the drug can cause unintentional, harmful and unwanted side effects. In some cases, such as with the drug lithium, the threshold between what is an effective dose and what is a toxic dose is very narrow. What is a therapeutic dose for one person might be toxic to another person. Drugs with a longer half-life can build up in a person's bloodstream and increase over time. Additionally, factors such as age, kidney function and hydration can affect how quickly your body is able to clear a medication from your system. This is why medications such as lithium require frequent blood testing to keep track of the levels of the drug in your bloodstream. The signs and symptoms of toxicity differ depending on the medication. Possible mild symptoms of acute lithium toxicity include diarrhea, dizziness, nausea, stomach pains, vomiting and weakness. More severe symptoms can include hand tremors, ataxia, muscle twitch, slurred speech, nystagmus, seizures, coma and in rare cases heart problems. Chronic lithium toxicity displays different symptoms including slurred speech, tremors and increased reflexes. Acute toxicity is more easily diagnosed as the symptoms will follow the one-time administration of a medication. Blood tests can also screen for levels of the medication in the person's bloodstream. There are several ways in which drug toxicity may be treated. If the toxicity is the result of an acute overdose, then a person may undergo stomach pumping to remove drugs that have not yet been absorbed. Activated charcoal may be given to bind the drugs and prevent them from being absorbed into the blood. Other medication may also be given as an antidote. Whether a drug will do more harm than good in an individual patient depends on many factors, including the patient's age, genetic makeup and pre-existing conditions, the dose of the drug administered and other drugs the patient may be taking. For example, the very old or the very young may be more susceptible to the toxic effects of a drug because of age-dependent differences in pharmacokinetic profiles or in drug metabolizing enzymes. Drug metabolism, genetic factors can alter how a patient metabolizes or responds to a drug. Therefore, individual responses can also occur because of genetic differences in drug metabolism or receptor activity as well as differences in the activities of repair mechanisms. While a spectrum of adverse effects may be associated with the use of any drug or drug class, it is helpful to conceptualize the mechanisms of drug toxicity based on several general paradigms. Number first, on target adverse effects, which are the result of the drug binding to its intended receptor but at an inappropriate concentration with suboptimal kinetics or in the incorrect tissue. Number second, off-target adverse effects which are caused by the drug binding to a target or receptor for which it was not intended. Number third, production of toxic metabolites. Number fourth, production of harmful immune responses. And number fifth, idiosyncratic responses. Now that we have understood about drug toxicity, let us now try to learn about pathology of drug toxicity. Drugs and their metabolites can interact with a diverse array of receptors to mediate adverse effects in vivo. Sometimes the parent unmetabolized drug causes toxicity, but often a metabolite of the drug reacts with the proteins, DNA and oxidative defense molecules such as glutathione to cause cellular damage and other adverse reactions. Drug toxicity can occur on many different time scales. Acute toxicity results from a single exposure to a drug with adverse effects resulting in minutes to hours. Examples of acute toxicity are the massive hepatic necrosis that can occur after a single toxic dose of acetaminophen. Many immune-mediated adverse effects occur within hours to days after administration of the drug. Chronic toxicity, on the other hand, refers to an adverse effect of a drug that occurs over a prolonged period of time. Long-term treatment with dopamine receptor antagonists for schizophrenia can result in tardive dyskinesia, an unfortunate on-target adverse effect that results from the critical role of dopamine as a neurotransmitter in the motor cortex. Hormone replacement therapy for postmenopausal women is another important example of chronic toxicity. Prolonged exposure to certain drugs or other metabolites can lead to fibrosis, organ dysfunction and birth defects as well as cancer. Now, let us discuss the next part of our today's deliberation, that is toxicity testing methods. Toxicology testing, also known as safety assessment or toxicity testing, is conducted to determine the degree to which a substance can damage a living organism. It is often conducted by researchers using standard test procedures to comply with governing regulations, for example, for medicines and pesticides. Much toxicology is considered to be part of the field of preclinical development. Stages of in vitro and in vivo research are conducted to determine safe doses of exposure in humans before a first-in-man study. Toxicology testing may be conducted by the pharmaceutical industry, biotechnology companies or contract research organizations. Toxicity tests examine finished products such as pesticides, medications, food additives such as artificial sweeteners, packing materials and air freshener or their chemical ingredients. The substances are applied to the skin or eyes, injected intravenously or intramuscularly or subcutaneously, inhaled either by placing a mask over the animal and restraining them, or by placing them in an inhalation chamber, or administered orally through a tube into the stomach, or placing them in the animal's food. Doses may be given once, repeated regularly for many months, or for the lifespan of the animal. Toxicity tests can also be conducted on materials needed to be disposed, such as sediment to be disposed in marine environment. Now, let us discuss the current methods related to toxicity testing. Toxicity studies are required to assess potential hazards to humans through the acute, subchronic and chronic exposure of laboratory animals to pesticides. The more specific types of toxicity that are determined include carcinogenicity, developmental and reproductive toxicity, mutagenicity and neurotoxicity. Detailed information on the metabolism or biotransformation of the pesticide is also obtained. Consideration is given to testing individual metabolites in animals and in or on pesticide treated plants to which humans could get exposed through their diet. The extent of metabolite testing required depends on the level of potential toxicity and environmental persistence of the metabolite. With the exception of the acute toxicity tests, more tests are conducted to determine the nature of any toxicity that can be produced by repeatedly dosing animals over an extended period. The results enable toxicologists to estimate the safety of a material of humans. Weil in 1972 published the following set of guidelines which reflected a consensus among toxicologists. These should be considered before initiating a toxicity test. Number first, use. Wherever practical or possible, one or more species that biologically handle the material qualitatively and quantitatively as similarly as possible to man. For this, metabolism, absorption, excretion, storage and other physiological effects might be considered. Number second, where practical, use several dose levels on the principle that all types of toxicologic and pharmacologic actions in man and animals are dose related. Number third, effects produced at higher dose levels are useful for delineating mechanisms of action, but for any material effect, some dose level exists for man or animal below which this adverse effect will not appear. This biologically insignificant level can and should be set by use of a proper uncertainty factor and competent scientific judgment. Number four, statistical tests for significance are valid only on the experimental units that have been mathematically randomized among the dosed and concurrent control groups. Number fifth, effects obtained by one route of administration to test animals are not applicable to effects by another route of administration to man. Next is acute toxicity studies. Acute toxicity studies provide information on the potential for health hazards that may arise as a result of short-term exposure. Determination of acute oral, dermal and inhalation toxicity is usually the initial step in evaluating the toxic characteristics of a pesticide. In each of these tests, the animal is exposed to the test material only once on first day. Together with information derived from primary eye and primary dermal irritation studies, also first dose on first day, which assess possible hazards resulting from pesticide contact with eyes and skin, these data provide a basis for precautionary labeling and may influence the classification of a pesticide for restricted use. Acute toxicity data also provide information used to determine the need for child-resistant packaging, for protective clothing requirements for applicator and for calculation of farm worker re-entry intervals. A minimum number of animals, usually adults, are used in these studies and only the endpoints of concern are monitored, that is mortality, observable skin or eye effects, dermal sensitization and observable neurotoxic behavioral changes. One exception is the inclusion of microscopic examination of neural tissues in the newly required acute neurotoxicity study. Now, let us discuss subchronic toxicity studies. Subchronic exposures do not elicit effects that have a long latency period, for example, carcinogenicity. However, they do provide information on the health hazards that may result from repeated exposures to a pesticide over a period up to approximately 30% of the lifetime of a rodent. Subchronic tests also provide information necessary to select proper dose levels for chronic studies, especially for carcinogenicity studies, for which an MTD must be selected. According to EPA, rats selected for these studies should be started on the test material shortly after weaning, ideally before the rats are 6 and in any case not more than 8 weeks old. For dogs, dosing should begin when they are 4 to 6 months of age and not later than 9 months of age. Next is chronic toxicity studies. Information derived from chronic studies is used to assess potential hazards resulting from prolonged and repeated exposure to a pesticide over a large portion of the human lifespan. These studies usually last 12 to 24 months. Of particular importance are long-term carcinogenicity studies, the purpose of which is to observe the test animals for the development of neoplastic lesions after a lifetime of exposure at dose levels up to and including the MTD determined from subchronic testing. And last is developmental toxicity studies. Developmental toxicity studies are designed to assess the potential of developmental effects in offspring resulting from the mother's exposure to the test substance during pregnancy. These effects include death of the developing organism, structural abnormalities, altered growth and functional deficiencies. Now, let us discuss the last part of our today's lecture, that is LD50 experiment. In toxicology, the median lethal dose, abbreviated as LD50, LC50 or LCT50, is a measure of the lethal dose of a toxin, radiation or pathogen. The value of LD50 for a substance is the dose required to kill half the members of a tested population after a specified test duration. LD50 figures are frequently used as a general indicator of a substance's acute toxicity. A lower LD50 is indicative of increased toxicity. The test was created by J.W. Treven in 1927. The term semi-lethal dose is occasionally used with the same meaning, in particular in translations from non-English language texts. LD50 is usually determined by tests on animals such as laboratory mice. The LD50 is usually expressed as the mass of substance administered per unit mass of test subject, typically as milligrams of substance per kilogram of body mass, sometimes also stated as nanograms, micrograms or grams per kilogram. Stating it this way allows the relative toxicity of different substances to be compared and normalizes for the variation in the size of the animals exposed. The choice of 50% lethality as a benchmark avoids the potential for ambiguity of making measurements in the extremes and reduces the amount of testing required. Lethal doses often varies depending on the method of administration. For instance, many substances are less toxic when administered orally than when intravenously administered. For this reason, LD50 figures are often qualified with the mode of administration. For example, LD50 IV. For disease-causing organisms, there is also a measure known as the median infective dose and dosage. The median infective dose is the number of organisms received by a person or test animal qualified by the route of administration. Because of the difficulties in counting actual organisms in a dose, infective doses may be expressed in terms of biological assay, such as a number of LD50s to some test animal. In biological warfare, infective dose is the number of infective doses per minute for a cubic meter. Now, at last, let us discuss the lethal dose 50 test procedure. A common form of the test is by oral dosing using a tube inserted down the animal's throat. Other forms of dosing include injection, forced breathing of the vapor and application of the substance to the animal's skin. Rabbits, rats, mice, birds and fish are the usual species, with dogs and monkeys also used. In the classical or formal LD50, several dose levels are administered to equal groups of male and female animals. An average of 60 animals may be used, although this would only be for one species and one particular dosing method. The test is allowed to proceed for 14 days, assuming the animals have not already died. Common signs of poisoning include unusual vocalization, tears, diarrhea, discharge and bleeding from the eyes or mouth and convulsions. No pain relief is given, yet the test must cause great suffering. Killing an animal during an LD50 test may invalidate the test results, since if left the animal might have lingered on for the 14 days or even possibly recovered and therefore counted as a survivor. The more animals that survive, the less toxic the substance is judged to be, with the corresponding commercial advantage of a lower hazard classification. As a measure of toxicity, LD50 is somewhat unreliable and results may vary greatly between testing facilities due to factors such as the genetic characteristics of sample population, animal species tested, environmental factors and mode of administration. That's all about today's lecture. Hope you have understood it well. Thank you very much and goodbye.